Today we're really going to cover kind of basic core structures that are not universal, but very common throughout large swaths, and we'll move into kind of more specific and particular function structures later on, or on Monday. Okay, so I wanted to start off with this question that I posed to folks at the end of class last time. So we talked about microscopy, right, and how we can assess how certain things work using different microscopy techniques. But something like imaging motility with a flagella posed a bit of a problem because on the one hand you need it to be alive, right? And on the other hand they're small. So how do you think, or do you guys have any ideas, maybe some of you even know, right? How did we accomplish being able to study something like flagellar motion in vivo or in live active organisms? Anybody have any guesses? So one hint is that the first time we did this was in an organism called E. coli. Anybody have any thoughts? Alright, everybody's shy this morning. So I'm going to try to show a video. Let's see if that jogs memory. What do you guys think?
Dark field.
Dark field. So dark field's a good guess, right? So you show it light from sort of either side, and that definitely has a dark background. But again, that thin structure would probably be something you couldn't resolve with dark field, right? And it looks a lot thicker there. So yeah, two hints. You've got a black background, and it looks a little different than, or it looks a little thicker, right, than that 0.2 microns. Do you have any guesses?
Is it DIC?
It's not DIC, yeah. One more black, what's the other black background microscopy technique?
Phase contrast?
No, phase doesn't have quite as dark. So we've ruled out a lot of them. what else is there?
Is it confocal?
Getting closer.
With fluorescence?
Fluorescence, right, exactly. So basically what people did is they engineered that flagellar protein with what you guys have probably heard of, a green fluorescent protein. Essentially one that sticks into sort of the pilling units but doesn't interfere with the polymerization of the flagella itself. GFP is a pretty big protein, and so it does make that flagellar appendage look pretty thick, but it also allows it to fluoresce, right? But what's exciting, and I'll show that video again, right, is you can start to get a sense of some of the unique dynamics of how this actually works, right? How, it's kind of this corkscrew shape structure, and depending on the rotation, right, that organism can move in either direction. Okay, so that's just kind of a way of people getting creative, right, using microscopy to solve problems that, sort of traditional microscopy doesn't necessarily allow you to do easily. All right, so again, so building off of microscopy, we're going to talk about, unique structures of microbes, ones that are commonly present throughout broad swaths of organisms. And this becomes important, right, because it's, these are the unique features of microbes that, we can think of for pathogens targeting antibiotics. If we think about our immune systems, these are things that our immune system target either for something that is pathogenic or something that's commensal, right, so something that we identify as a friend as well. And it's also important for these organisms, for phage in particular, right, something that, I'll talk a little bit about later on in the course that we utilize a little bit more commonly as targets for particular microbial pathogens, but a lot of these structures are also targets for phage infection, and so are therefore important in that way. And we're also going to talk a little bit about some of the capabilities that these structures infer in organisms. All right, so I talked a little bit about the organisms being big last time and how there are a few sort of gargantuan, gigantic microbes. This happens to be one of my favorite ones, Epilepicium. When I was a graduate student, I rotated through a lab that did work on this megabacteria that you can actually see with the naked eye. So we don't have this organism in culture, so the graduate students would get samples of fish guts from sturgeon fish. So poor grad students, they had to go to the tropics and collect fish. But they would have to, in order to study these organisms, extract those microbes from the gut, and you could actually sort of like pick them out with a pipette tip using a dissecting scope, which is not something most microbiologists can do. So they are visible by the naked eye, and they are just incredibly huge. So just this is a size comparison, right, from a normal E. coli versus this Epilepicium, even a micro-eukaryote here, a paramecium. So they have about 2,000 times the volume, and so this organism just has to do a lot of things differently. It has tons and tons of copies of its genome, I think on the orders of tens to hundreds of thousands. And it does this kind of weird cell division, so rather than replicating by binary fission, it actually forms daughter cells within the actual structure. And then has kind of an alien moment, right, where the daughter cells kind of burst out, and kind of gruesome. But, all right, so first PRS question. So which is a possible selective advantage of being large if you are micro? So you can store more food, it's less energy, you can avoid predation, or you can outcompete smaller organisms. Excuse me? Yeah.
What's the session ID of Longview?
Yeah, it's
971638.
Thank you. 71638. I really hate that this room doesn't have a chalkboard. Sorry, I thought I put it on all the slides, but I must have missed this one. Just come talk to me after class if you had a problem. All right, so as most people said, right, you can store more food or energy, right? And for organisms that we talked about, like the Thiomicrospira, right, this is a really important one. If you live in sort of energy unstable environments. Avoiding predation, a lot of predators for microbes are things like viruses. I don't know that that would necessarily give you a benefit. I guess if you were getting eaten by paramecium or something like that, being bigger could be a benefit. Or you can outcompete smaller organisms. And we talked about how this last one in particular, right, is probably not the case because of this difference between surface area to volume ratio. So really the main focus one, or at least the one we talked about, right, is storing more food or energy. All right, so we're gonna move on to talking about the cell envelope. And all right, so the cell envelope is essentially everything that includes sort of the outermost barrier or the surface of the cell. So including the cytoplasmic membrane, all of the stuff that goes on outside of that cytoplasmic membrane. And so we're gonna start off by, okay, so yeah, everything technically that is outside the cell or encompasses that outer barrier of the cell. All right, so we're gonna start off by talking about which structures or things involved are universal to all microbial cells. So cell walls, cytoplasmic membrane, mitochondria, or flagella.
So eukaryotic cells, we learned that they have like I think it's called a extracellular matrix or like cloud proteins and stuff around them. Is that analogous to the envelope in bacteria?
It probably would count as part of the cell envelope, but usually when we're talking about this, we're talking about single-celled organisms. And so a lot of those structures are kind of
They
more,
connect to other cells.
yeah,
Yeah.
more kind of tissue specific or multicellular organism specific structures. All right, yeah, so as most people said, right, the cytoplasmic membrane, right, is this universal feature. So we'll talk about organisms that do not have cell walls that are still microbial. Obviously, mitochondria are going to be specific to organisms that are eukaryotes, and flagella even are not the only way that organisms accomplish motility, and certainly not all organisms are motile. All right, so the cytoplasmic membrane, right, it's this kind of core barrier to what makes the inside of the cell versus the outside of the cell. Classically, in bacteria, this is composed of phospholipids, essentially fatty acids that have this hydrophobic chain unit. You guys have probably all seen this many times from intro biology all to cell biology, and so this is something that's similar, right, between eukaryotes and bacteria. How it gets supported, a few little details, things like chain lengths, degree, the ability to modulate sort of the degree of the essentially degree of saturation vary across even species of microbes. But the idea is fundamentally the same, right? So you have this hydrophobic core, right, which is going to repel water, that is surrounded by these sort of hydrophilic moieties on either side that make kind of naturally a sort of a vesicular structure. But it's not an it's permeable, right? It's not an impermeable barrier. So, again, these moieties are not tethered together, right? So there's no structure that keeps them kind of in direct sync with each other. And so movement, stretching, they're essentially forces can act on them, right? And things like water and small molecules can and do diffuse through them. And so this essentially is kind of one of the main features here for these fatty acids. Another thing that I want to just kind of emphasize, because this is going to be a difference when we talk about bacterial and archaeolipids, is that one of the key features here between the prokaryotic, or sort of the bacterial, and the eukaryotic ones, is that you have this sort of ester linkage between a fatty acid chain here and that this phosphohead unit. And again, so that's something that's a little bit different. So archaea do it differently. Again, all of these sort of lengths within that fatty acid, the branching patterns, all of these things can and do vary. A lot of this has to do with the type of habitat that this organism might live in, right? So fluidity is going to be something that is going to change depending on the temperature of the system you live in, right? The salinity of the system you live in. And so organisms have the ability to sort of modulate these sort of fatty acid structures to help overcome those differences or those environmental parameters. We can and do use these also as sort of a fingerprint that let us know about particular microbes. So differences, broad classes of differences might tell you whether or not something falls within a particular group of organisms. We'll talk a little bit about hoponoids later. So presence of specific sort of functional units might also help us, maybe not identify species per se, but at least give you a rough idea of what that organism is. And for certain organisms, it might even tell you what sort of environmental stresses they're exposed to. So one thing that is also kind of important, or can be important within sort of that lipid matrix, is that there are other sort of aliphatic compounds that organisms produce. You guys are probably familiar with sterols that are made by eukaryotes, right? This is thought to be sort of the main membrane strengthening unit that eukaryotes have, and it's very specific to eukaryotes. However, bacteria also make sort of similar structures called hoponoids, which functionally have a very similar purpose, that they serve essentially to help strengthen membrane integrity. These are something that are, again, not found in archaea, right? So this is another difference sort of a similarity, right, between the membrane structures that we see between bacteria and eukaryotes. All right, so archaea, on the other hand, so just to kind of emphasize that overall basic structure that we're seeing of membranes is universal for cytoplasmic membranes throughout the bacteria and also within the eukaryote. There are, again, some differences in those fatty acid structures and lengths, and probably also some of those stabilizing molecules like steroids and sterols and hopanoids. But when you get to the archaea, lipid diversity is just immense. So one of the first things to point out is that the linkages that are made between that head unit and the fatty acid chain are now no longer ester linkages, they're ether linkages. These are slightly stronger, they're a little harder to hydrolyze, so it's thought to be one of the potential reasons why archaea may have adopted these types of lipids. There's sort of a classic model that archaea are extremophiles, which is not true. There are lots of mesophilic archaea that are very important in lots of environments, and there are even extreme environments that don't have archaea. But again, these lipids seem to be more robust, at least in their tethering to something like the glycerol side chain here. There's, again, a couple of different moieties here. Another thing that can be kind of distinct is that in some archaeal species, you now no longer have a lipid bilayer, but you have essentially a monolayer that has more or less the same overall structure. And the way this happens is you now have two sort of phospholipid head groups that are tethered by that hydrophobic cord chain. And so, the difference here, right, is that, you're not going to have these sort of separate sliding entities within the sort of inner and outer membrane. So these essentially hydrophilic head units are going to be tethered together. And we're not exactly clear, I think, what the consequences of this are, but it seems to be a pretty common adaptation in the archaea. Another distinction, right, is that these units here are isoprenoid in structure, and so instead of having sort of an acetyl fatty acid group, you now have these isoprene units. So in general, these are quite a bit different than what we see for the bacteria. All right, so again, just to sort of emphasize these main points, overall, the membrane sort of integrity and structures probably don't vary too much, right? So they're going to have access to sort of the same, similar intermembrane proteins, similar activities that go on at the cytoplasmic membrane. But again, some of these chemical details are what functionally vary between the two. All right, so a quick question, right? So bacterial membranes are more similar to archaeal membranes, than eukaryotes, true or false? Everybody was awake for the last few minutes. Great. So yeah, it's false. And this is somewhat surprising. We'll talk about this a little bit when we get to evolution, because the sort of dominant hypothesis right now is that eukaryotes actually stemmed from an archaeal lineage called the Asgard archaea. And one of the major unresolved questions in that is, well, where did they get these phospholipid bilayer membranes that look exactly like bacteria? And to this date, I don't think we have a good answer for that. All right, so again, this is a membrane that has to selectively allow things through. We talked about how naturally it's not, it's a permeable, but it's not, it doesn't let every old thing through, right? So there's a preference for things like small molecules, and or things that potentially have some, again, aliphatic characteristics that have some hydrophobic entities to them. But cells need to be able to regulate, right, what goes in and what goes out. And so the cytoplasmic membrane is much more than just sort of this membrane structure, right, this lipid bilayer. It's studded with all kinds of proteins, and as I'll talk about in a minute, has a lot of other structures associated with it. But, right, so one of the key sort of features of cytoplasmic membranes, especially in archaea and bacteria, unlike eukaryotes, right, this is where all of sort of the energy metabolism happens, right? And so part of this role is not just being able to keep solutes concentrated inside versus outside, but you also have to sort of maintain a charge barrier, right? That's going to allow you to do work, right? To make things like ATP, to import different solutes, etc. And so there's a lot of basic roles that this cytoplasmic membrane has to do. It's got to have these proteins that are capable of transport, but also that provide anything that needs some sort of structure. You have these proteins that are anchored in there as well. All right, so from your book, you talked a lot about active transport. So what are they? What is the energy source or energy sources used for active transport? ATP hydrolysis, proton motor force, energy coupling, and/or all of the above. So most people said all of the above, right? Which is right. So it just depends on what you're trying to get in the cell and what that transporter sort of evolved as a mechanism in order to do that, right? So certain ions have sort of antiporter systems where you can import an energetically favorable one and export an energetically favorable, an endergonic one or one that's not energetically favorable to export, and vice versa. Right, so it doesn't necessarily have to be protons, but that is one that is incredibly commonly used. And for certain things, you have to use a little bit more energy, so we tap into something like ATP hydrolysis, which can carry, a couple of protons worth of energy rather than just a single proton. All right, so this is just kind of an overview, right, of some of the different transporters that get utilized. Again, emphasizing that point, you can have different ions that get coupled together, ions coupled with, again, these are sort of different antiporters, synporters, things that specifically target molecules and use ATP hydrolysis to bring them in. And or, again, this is an example of just a coupled reaction that essentially uses the first really energetically favorable step, or a step that hydrolyzes ATP, produces glucose, 6-phosphate, to essentially drive the energetic favorability of importing glucose here. So there's a lot of, these are just kind of general mechanisms. There's a wide variety of things that microbes have to get in, right? So not every organism even just uses glucose. Could be any carbon source. If you don't use carbon, it could be your sulfur source, nitrogen source, etc. So anything that doesn't freely diffuse into the cell, you have to invest energy into bringing it in. Again, this kind of harkens back, right, to where that surface area to volume ratio problem comes in, right? So you have you need more of these and more active ones of these if you have to support sort of a larger volume to get your processes to go. All right, so next we're going to talk about cell walls. Again, you guys have probably talked about cell walls in cell biology, right, with the concept of plant cell walls. So certainly some eukaryotic organisms have different cell wall types, right? You have cellulose-based ones in plants, you have chitin ones and things like fungi. But they're slightly different in bacteria, and most bacteria and archaea, some again microeukaryotes, most bacteria and archaea have cell walls. So not all of them. There's a few oddballs in the world, always. But the predominant idea here is that essentially we talked about how that membrane, right, allows a little bit of transfer, and it's not in and of itself structurally well tethered together, right? So if you have huge pressure differentials, it's relatively easy for that membrane to prop. And so people have done experiments, for example, with E. coli, where if you get rid of the cell wall, in most solutions, that cell just kind of grows and grows and grows until it explodes, right? Because it has this high concentration of things like sugars, et cetera, in its cytoplasm, and it's fighting against all that water rushing in. So essentially the cell wall is to protect or help support the cell structure under those great osmotic or turbo pressures. And prokaryotes have a pretty amazing ability to withstand really high pressures. So I've done a lot of work in deep sea microorganisms, and I always find this absolutely amazing, but you can take microbes from sediments hundreds of meters of depth and bring them to the surface, and they don't all explode. I don't know how. That's an amazing amount of pressure differential that they're dealing with. And so part of the answer is that they're pretty well adapted to dealing with pretty incredible osmotic pressures. And so this cell wall, which we'll talk about, seems relatively simple, but it's actually really, really robust. And so we're going to focus on sort of the most common cell wall type, which are gram-positive and gram-negatives. These are sort of the first that were identified and really well studied. And the key differences, right, as we talked about last time, are the actual cell wall thickness itself. So gram-positives have this incredibly thick peptidoglycan layer. We'll talk a little bit more about what peptidoglycan is in a minute. And this allows them to absorb more of that crystal violet into their cell wall. This is a polymer that's really very similar to starch, which is also something that crystal violet reacts with. And in gram-negatives, you still have a thick, complicated cell envelope, but the peptidoglycan point, right, that essentially starch-like structure that's making a cage around the cell, is much thinner. And so because of that, it's easier to sort of remove the staining of crystal violet when you have this differential assay, the gram stain. All right. Right, so gram-positives, thick cell wall. You've got some other things like teichoic acids, lipoteichoic acids. And another key feature is you only have that cytoplasmic membrane, whereas in gram-negative bacteria, you actually have two membranes. So there's a separate membrane outside of the cell. So in some cases, this acts as sort of a double selectively permeable barrier. And we'll talk a little bit about this later, but there are sort of larger openings in general in that outer membrane, things like porin structures and or sort of bigger gaps to allow, make it so that you don't need active transport in both the outer membrane and the inner membrane. But you also have sort of a lot more structure on the outside that allow for sort of these lipid-protein interactions, something what we call LPS, which we'll talk a little bit more in more detail about later. But all right, so most bacterial cell walls are this peptidoglycan structure. Again, looks very similar to starch, right? With the exception that the the backbone here is not going to be just glucose, but is rather going to be this N-acetylglucosamine or N-acetylmuramic acid. So these are both things that relatively easily interconvert from glucose. So one of this, I guess, reasons why this might have efficiently evolved is because if you're an organism eating things like starch, you have enzymes, right, to metabolize starch. If you have a lot, right, it makes sense to kind of make similar building blocks of something you have a lot of resources for, but that aren't going to be reactive to the enzymes that you have. And so a lot of these molecules have kind of structures that are attached to places where an enzyme might normally break down that polymer, right? So they've co-opted, part of their building blocks, but made them really more robust in general, or less amenable to breakdown. It's also given them places or little footholds for doing other things that allow interlinking, right? So a lot of starch polymers are basically just a long chain of sugar acids or sugar molecules, and essentially, right, you want more of a cage-like structure. And so these moieties here also allow for places where you can start to do kind of cross-linkages. So this is, I would say this is an overall, again, general structure, but just like with fatty acids, there are nuances across bacteria, little things that get changed, little cross-linkages that get modified, etc. Most of them can still be destroyed enzymatically, and we'll talk a little bit about this later, about the enzyme lysozyme in general. So there's still some sort of general structure going on here. All right, so again, we talked about how there's different, essentially, appendages here that you can build off of and allow cross-linkages. These become important in part because, again, parts of these are also picked because they become challenging to try to degrade. We have, for example, a racemarized amino acid, an L amino acid versus a couple of D amino acids that get utilized in this functional chain, which again just makes them a little bit harder to break down for normal cells. All right, so beta-1,4 linkages are pretty strong. So they're again similar to something like glycogen and cellulose having these sort of alpha or beta-1,4 bonds. So these are again very common in other polysaccharides. All right. So, but one of the things that the reading points out and what we'll talk about is that antibiotics are good targets for killing bacteria. And so what are some of the reasons for this? And just, if you have a clicker, I think if you enter one, it'll still give you, I apologize, I'll try to mimic these in the future. Say my name more time. All right, so this is kind of, a bit of a weirdly worded question, right? So, but, why are antibiotics such good targets for killing bacteria? So, I'm sorry, in my head that meant to be, why are cell walls, antibiotics that target cell walls such good targets for killing bacteria? So, yes, absolutely, weakening the cell wall. A lot of antibiotics target cell walls. And that's just, a pretty natural phenomenon that we have observed. And you're right, so if you damage the cell wall, that's super bad for the cell, right? But there are a lot of critical life processes, right? So why did antibiotics, or why has it been more common, right, for antibiotics to favorably interact or weaken cell walls? And part of the answer has to do with the fact that, well, if you're impacting the thing that's on the very outside, right, you don't have to worry about actually getting into the cell and actually to have your mode of action. And then that tends to be one of the biggest barriers to antibiotic development is, you can make something that's really toxic or harmful to the cell, but getting across that cell envelope is not trivial, right? Again, size is important, so if you have big old molecules, those aren't going to easily get into the cell, and there aren't always great vectors for getting antibiotics into the cell that